Welcome. An individual was found to be of blood group B plus. Name the two antigens present in his where blood cells. That's the red blood cells. And when you check, it's B plus. So it means the person has a rhesus antigen. And because it's a B plus, the first antigen should be antigen B, then antigen D for the rhesus. So we are speaking about what? Antigen B and antigen D. Adaptations of the alveolus. 3 marks. Number 1, moist. Number 2, thin epithelial membrane. Number 3, highly vascularized. It's highly what? Vascularized. And when you speak about being moist, what's about this one? Because the question reads adaptation. You will not get any mark when you just live in that manner. To dissolve the respiratory gases. Which to diffuse in what? Solution form. To diffuse in solution form. That is the first. Thin membrane to reduce diffusion distance. This one reduces what? Diffusion distance. For faster what? Diffusion. Highly vascularized is just to transport the respiratory gases. Respiratory what? Gases. That is it. It's 3 marks. What's oxygen date? It's extra amount of oxygen required to get rid of accumulated lactic acid when the oxygen supply is less than the demand. That is it. So from this point now, we discuss this other one, which is about what? You are given a question here, and the question reads, the setup below was done by the same student to investigate a certain physiological process. We are speaking about what? It was done by some students to investigate a certain biological process. Identify the process. So you can check, we have oil layer, we have boiling tube, you have glucose solution, and the yeast. So this process is respiration, but now you have oil layer, so it means there is no entry of air. And which kind of respiration? It's anaerobic respiration. Anaerobic respiration. Then you're being told, you give a question for the reaction in the boiling tube. For the reaction in the boiling tube. So you're speaking about here. You have glucose, which is being broken down in absence of what? But now we have the yeast. So yeast is an enzyme, and you will get what? Yeah, the release of carbon 4 and also ethanol. So it will be what? Glucose, which is the solution, in the presence of yeast, which is an enzyme, and I'm saying you get ethanol plus lime water will change from a white precipitate because of the presence of carbon 4 oxide, which is carbon 4 oxide, and also water will be released. Speaking about what? Water being released. How can the rate of product formation in the boiling tube being increased? How can we increase the formation, rate of product formation in the boiling tube? Number one, you increase the yeast. You increase what? The yeast. Number two, because of these again, you can ensure that the temperature, is at optimum. Ensure your temperature is at what? Optimum. Ensure your temperature is at optimum. And also you can still increase the glucose concentration of the solution. That is the raw material for the respiration. You increase the yeast, so the reason for increasing the yeast, it's like you're increasing the enzyme concentration, which will break down the glucose concentration. So let's go to this other question. The question below represents a role of a certain organ in mammals of a body. You can see this NH2. What's NH2? That is amino group. And you see there is an addition of an hydrogen ion. You form ammonia. Ammonia is being broken down still in the presence of carbon 4 oxide. We get urea plus water. Which process is this? This process is deamination and not detoxification. If we start from this point going forward, it will be detoxification. But when you go on this other end, NH2 is amino group. And how do we attain the amino group? Amino group is attained through the breakdown of ammonia. And ammonia is being broken down to amine group, that is the amino group, plus carboxyl group. The carboxyl group can be oxidized to release energy or glucose. Then we have this now amino which combine with hydrogen and we get ammonia. So we are speaking about the process here is what? Deamination. Organ, where does this process take place? It's taking place in the liver. Liver is our main organ. Then you're being told from an extension of the same question, apart from process named in B above, that's deamination, state two other roles of the organ. That is the roles of the liver. You can speak about detoxification, blood sugar regulation. You can speak about thermoregulation and many more, like even our formation of the red blood cells in infants, protein synthesis, synthesis of vitamin K, and many other more. Thank you very much.